The fourth module is all about the risks and benefits of water reuse projects and how these apply to project implementation. In the first section, we're going to look in more detail at health risk management under the World Health Organization framework. First, we'll define some key terms that will help us understand the framework for risk assessment and management. Then we'll look at a useful resource developed by the WHO called the Sanitation Safety Planning Manual. Definitions and risk framework. All activities that we are involved in carry some degree of risk. Risks can either be voluntary, such as cigarette smoking, or involuntary, such as breathing air polluted with car emissions or drinking water contaminated with carcinogenic chemicals. We've also seen the word hazard mentioned in reference to the HACCP principles, or hazard analysis and critical control points. So what's the difference between a hazard and a risk? A hazard is a biological, chemical, physical, or radiological agent that has the potential to cause harm, for example, the bacteria that cause cholera. A risk is the likelihood of a particular hazard causing harm in exposed populations in a specified time frame and what the magnitude of that harm would be. The risk assessment is the process undertaken to evaluate whether there is a risk and, if so, how severe it is. And risk management incorporates understanding, evaluating, and prioritizing risks for a given system and then implementing appropriate risk reduction strategies. During a risk assessment, potential hazards need to be identified and characterized, usually according to a dose-response relationship, to see how hazardous it is. Next, it's necessary to assess the level of exposure of a population to a certain hazard under different conditions. All of this information is consolidated to characterize the risk in relation to its potential impact compared to other risks. This produces the overall risk assessment. As you'll recall from Section 3, it is the Stockholm Framework for Water Quality that is used in the WHO guidelines. It uses such a health risk assessment approach to develop health-based targets for water reuse that can be generalized to different contexts to a certain extent. The data used in the risk assessments in order to develop the guidelines include microbial and chemical laboratory analysis, epidemiological studies, and QMRA, or quantitative microbial risk assessment. This resource helps guide project leaders so that these time and cost-intensive studies don't have to be carried out each time. It should be noted that currently we have a better understanding of microbial health risks in comparison with chemical health risks. This is due to the fact that there are thousands of synthesized chemicals that are used but that have not yet been studied to determine their short-term or long-term health impacts. Therefore, most of the risk management framework tends to be focused on the microbiological risks, as they are the most well-known. It is expected that modifications to health risk management may be implemented as more information about chemical risks becomes available. The level of health protection required is going to depend on the nature of the hazard and which measures can be put into place to minimize risk. While the general public may prefer the idea of zero risk, in a world of limited resources and competing demands, the idea of tolerable risk is vital in order that health targets are sensible and achievable and that measures to pursue them are cost-effective. There is increasing recognition, especially among policy-making and scientific communities, of the concept of acceptable risk. The term tolerable risk is preferred by some in recognition that the risk is not truly acceptable but may be tolerated, in deference to greater or more highly perceived priorities. The standard metric of the impact on health due to a hazard or disease is the Disability Adjusted Life Year, or DALY. DALYs are a measure of the health of a population or of the burden of disease due to a specific disease or risk factor. They attempt to measure the time lost due to disability or death resulting from disease, compared with an extended life free of disability and disease. DALYs are calculated by adding the years of life lost due to premature death to those lost due to disability. Years of life lost are calculated from age-specific mortality rates and standard life expectancies in a given population. Years of life lived with a disability are calculated by multiplying the number of cases by the average duration of the disease. These figures are standardized by a severity factor depending on the disease. The DALY concept is an important tool for comparing health events, as it takes into account not only acute health effects, but also delayed and chronic effects. The standardized target is 10 to the minus 6 DALYs, or 1 disability-adjusted life year lost per 1 million life years. When risk is expressed in DALYs, we can compare different health events, for example, diarrhea, typhoid, and E. coli, and assign priorities to risk management decisions. The risk assessment and estimation of DALYs is the basis for the water quality values presented in the WHO guidelines. The flowchart presented here is a visual representation of the Stockholm framework used for developing the harmonized guidelines for the management of water-related infectious diseases. It shows the relationships between the concepts we've seen, including health-based targets that come from the assessment of health risk associated to environmental exposure, and the idea of a tolerable level of health risk. The health risk management actions must be well defined, and they will depend on the definition of water quality and other objectives. While water quality targets may be expressed in terms of exposure to specific pathogens, care is required in relating this to overall population exposure, which may be concentrated into small periods of time, such as during disruptions to normal operations, such as equipment failure or during a storm, or even large-scale outbreaks of disease. The possibility of short-term events leading to very serious health events provides a logical justification for the multi-barrier principle in water safety. This concept looks at all the components of a system and identifies safeguards at different points throughout in order to provide security of performance and a certain level of redundancy. It's also used in recognition that there are many different measures that can be taken to achieve a similar level of pathogen reduction, and they don't necessarily involve costly treatment equipment. This is important for communities that do not have large and secure budgets to invest in infrastructure upgrades. The objective is to reduce the risk to an acceptable level and/or to minimize risk by optimizing the risk reduction throughout the system and by optimizing the available barriers. If we look at microbiological health risk, we can see the typical load of the 4 main types of microbes: bacteria, viruses, protozoa, and parasites. There are many microbes that are hazardous to humans. However, we don't develop risk plans for all of them, and we don't routinely test for all of them in water samples. Over time, it has been seen that certain microbes can be used as reference pathogens for groups of pathogenic microbes. Then, if we can treat or manage the risk of the reference pathogen, it is very likely that we will also have addressed risks related to a number of other similar pathogens. Hence, the microbes shown on the screen are some of the common ones used as such reference microbes. We can also see that the numbers of these pathogens in wastewater are extremely large. There are 2 methods used to set health risk limits in relation to microbes. The first is simply to set a numerical limit on the number per unit volume, as seen here for fecal coliforms in the WHO guidelines. In this case, the irrigation of crops likely to be eaten uncooked should be done with treated wastewater with less than 1,000 fecal coliforms per 100 milliliters of water. If we look at a chart of the typical pathogen content of wastewater, the same values as we saw before, we can get an idea of how much treatment is necessary to achieve the desired concentration of less than 1,000 or 10 to the power 3 fecal coliforms per 100 milliliters. If we suppose that there are 10 to the power 7 or 10 million fecal coliforms per 100 milliliters in the wastewater entering the treatment plant, that means it's necessary to reduce the load by 10,000 or 10 to the power 4. Or we can say it's a 4 log reduction. And this brings us to the second type of pathogen target or health limit that is commonly imposed. It is a reduction in pathogen load, sometimes regardless of the initial or final pathogen load. These two types of limits are often used in combination. Pathogen reduction targets can be seen in Table 4.1 of the 2006 WHO guidelines, where rotavirus reduction is specified for different crops using different irrigation methods. For example, a greater reduction is needed for onions, a 7 log reduction, compared to lettuce, a 6 log reduction, when irrigating without any restrictions to the crop or to the workers. One of the benefits of thinking in terms of pathogen log reductions in comparison to simply using a target value is that typical log reduction values can be assigned to many different treatments and control measures, and these log reductions can be added together to achieve the desired reduction total. Examples of typical values are shown in Table 4.3 of the WHO guidelines. Adding the log reductions from different control measures together is the basis for the multi-barrier risk management approach and is a key part in the development of a comprehensive risk assessment and risk management plan. Some standard or typical pathogen reduction values for control measures may be reasonably fixed, but others can depend on the specific situation as shown in the notes column of the table. Pause the video to read in detail or find the table in the guidelines. We saw this chart in Section 3 on regulation. The multi-barrier management actions or health protection control measures are illustrated. Example A shows a 4 log reduction from treatment, a 2 log reduction from die-off, and another 1 log reduction from washing the produce. This combination of measures provides a 7 log reduction overall in order to achieve the health-based target of less than 10 to the negative 6 DALYs per person per year. Before we move on to the next section, it's worth noting the difficulties that many people have with thinking in exponential or logarithmic terms. If we compare these values of pathogen loads on a logarithmic scale compared with the same values on a normal linear scale, we can immediately see the difference in the orders of magnitude between the viruses and all the other types of microbes that makes the linear scale not particularly useful in this context. If we remove the viruses from the linear chart and resize the vertical axis, we can see a little better the remaining pathogens, but it's still not so helpful. Again, if we remove the total coliforms from the linear chart and resize the vertical axis, it's still not so helpful, hence why we use logarithmic scales. The extremely large numbers that are involved when we use logarithmic scales are known to be very difficult for the human brain to work with. Hence, it can take some time to get used to working with log reduction values and to have a sense of what they mean practically. For example, a 99.9% reduction in pathogen load may seem great, but it's only a 3 log reduction. A 7 log reduction for viruses, for example, requires removal of 99.99999% of the viral load. To summarize, at its most simple, waterborne disease risk management involves identifying potential sources of contamination and managing barriers to prevent contamination reaching end users. In an ideal system, this would be satisfactory since all scenarios by which contamination could enter the system would be understood, barriers would be effective at eliminating the risk from these sources, any barrier failure would be detected and corrective actions taken immediately, and individuals with the power to manage risk would have this as their primary interest and would behave appropriately. In reality, the arrangement of waterborne contamination sources and barriers is very complex and is never perfectly understood. Barriers are rarely absolute barriers and function primarily to reduce risk, not to eliminate it. Finite resources limit the ability of contamination sources to be controlled at source or via barriers, and individuals with the power to manage risk may have conflicting interests, and people cannot be controlled and relied upon totally. Hence, it's important to have a realistic expectation for your risk management plan, all the while striving to improve the operations. Additionally, it was noted in Section 3 that there was a shift in the emphasis that occurred in the WHO guidelines from the 1989 edition to the 2006 edition. Some criticisms of the 2006 guidelines is that they are too academic and technical to be useful for many contexts in developing countries. Being aware of these challenges, the WHO developed the Sanitation Safety Planning Manual, which has helped to operationalize the 2006 guidelines. Let's now look at the Sanitation Safety Planning Manual and related resources by the WHO. The manual itself is available for download from the WHO website. It was first published in 2016 and an updated second edition was released in 2022. It takes project managers through a logical series of modules where you'll need to prepare for the sanitation safety planning activities, describe the current and future sanitation system, identify hazards and hazardous events, and assess existing control measures and exposure risks, develop and implement an incremental improvement plan, monitor control measures and verify performance, and develop supporting programs and review plans. The manual contains practical examples for a variety of different contexts that you can follow step by step to get an idea of how the process could function in your context. It also provides guidance on how to use certain tools, such as excreta flow diagrams, in order to better identify priority parts of your sanitation system for improvement measures. Another tool to help prioritize within your system is the SaniPath Exposure Assessment Tool. It provides guidance for standardized primary data collection. The data are then used to automatically produce an exposure assessment analysis. In order to assist in using the manual itself, there is also a Learning Hub website that contains training materials, PowerPoints, and worksheets, as well as fact sheets and self-study videos on each of the modules of the manual. This resource is targeted at a variety of users at different levels, including health authorities and regulators, local authorities, sanitation utility managers, sanitation enterprises and farmers, community-based organisations, farmers associations, and non-governmental organisations. It can be a useful tool to help standardise the process of assessing risk and developing management plans for your current and future water reuse projects.